Welcome to this lesson on nonsense mutations. Nonsense mutations are a critical type of genetic mutation that can have severe consequences for protein production. In this video, we'll explore how these mutations create premature stop signals in our genetic code, leading to incomplete and non-functional proteins. Understanding nonsense mutations is essential for grasping how certain genetic diseases occur at the molecular level. A nonsense mutation is a specific type of point mutation, meaning it involves a change in just a single nucleotide in the DNA sequence. What makes it particularly significant is that this single change converts a normal codon that codes for an amino acid into a stop codon. Stop codons are signals that tell the cellular machinery to halt protein synthesis. When this happens prematurely, before the protein is fully built, we get a truncated or shortened protein that typically cannot perform its intended function in the cell. In the genetic code, there are 3 stop codons that signal the end of translation. These are UAA, known as Ochre, UAG, called Amber, and UGA, known as Opal. Normally, these stop codons appear only at the end of a gene to signal that the protein is complete. However, in a nonsense mutation, one of these stop codons appears earlier in the sequence due to a point mutation, causing premature termination of the protein. Let's look at a concrete example to understand this better. In a normal messenger RNA sequence, we might have a UG, UCU, CAG, UUC, GGA. This sequence would be read continuously to build the protein. However, if a nonsense mutation occurs, the third codon CAG might change to UAG, which is the amber stop codon. Notice that only one nucleotide changed from C to U, but this single change has dramatic consequences because UAG signals the ribosome to stop translation immediately. The impact on the final protein product is striking. With the normal sequence, translation produces a complete protein chain: methionine, serine, glutamine, phenylalanine, and glycine. However, when the nonsense mutation is present, translation stops prematurely at the new stop codon. The result is a drastically shortened protein consisting of only methionine and serine before the ribosome encounters the stop signal and releases the incomplete chain. This truncated protein is almost always non-functional. Nonsense mutations have serious consequences for cellular function. First, they produce shortened, non-functional proteins that cannot carry out their normal roles. Second, these truncated proteins lack critical protein domains, which are specific regions necessary for the protein's function. Think of it like building a car but stopping halfway through. You might have the chassis and engine, but without the wheels and steering system, it cannot function. Third, because proteins are essential for virtually all cellular processes, nonsense mutations often cause severe genetic diseases. Many well-known genetic diseases can result from nonsense mutations. Cystic fibrosis, a disease affecting the lungs and digestive system, can be caused by nonsense mutations in the CFTR gene. Duchenne Muscular Dystrophy, which causes progressive muscle weakness, often results from nonsense mutations in the dystrophin gene. Beta-Thalassemia, a blood disorder affecting hemoglobin production, can arise from nonsense mutations in the beta-globin gene. These examples demonstrate how a single nucleotide change can have profound effects on human health. To summarize, nonsense mutations create premature stop signals that halt protein synthesis before the protein is complete. The result is truncated, non-functional proteins that cannot perform their intended roles in the cell. This loss of protein function is the underlying cause of many genetic diseases. Understanding nonsense mutations helps us appreciate the precision required in genetic information and why even small changes at the DNA level can have such significant consequences for an organism's health and development.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.